Earth is teeming with life. From tropical rainforests to concrete jungles, life can be found almost anywhere on this pale blue dot we call home. However, for the longest time, this was not the case. 4.5 billion years ago, Earth was inhospitable. Volcanoes spewed clouds of ash and smoke into the sky, while streams of lava gushed from their craters. For 300 million years, the young planet was constantly bombarded by asteroids and meteorites in a period called the Late Heavy Bombardment. Yet, in these unlikely conditions, perhaps in a warm little pond, as speculated by Charles Darwin, chemistry made the leap to biology. How it happened remains a mystery to this day. The Australian Centre for Astrobiology, based at the University of New South Wales, is the only centre for astrobiological research in Australia. In this program, we showcase the work of young scientists in the ACA in tackling the leaps of complexity that eventually led to life as we know it. In order to understand how early life emerged, one must ask, what were the chemicals present? While much of the early Earth's chemical inventory came from the planet itself, a significant portion arrived from extraterrestrial bodies. The extreme conditions of space led to the formation of many compounds not easily synthesized via conventional chemistry. To understand the mechanisms by which these chemicals form, astrochemists like Zach Levy use a technique called spectroscopy.
Spectroscopy provides a means to study interstellar environments that are currently unavailable to humanity. By investigating how light interacts with molecular clouds, which are the nurseries for new star systems, allows us to gain an understanding of the molecular catalogue that was available when our solar system was first formed. Comparing interstellar data with laboratory spectra of gas phase species helps to confirm the existence of these molecules in space. My research focuses on the formation of a class of molecules called polycyclic aromatic hydrocarbons, which may be the cause for a series of interstellar absorption features that we observe towards stellar objects. These compounds, which are ubiquitous across the universe, may give us a hint to the ingredients of the primordial soup from which life emerged.
Cells are the basic units of life, comprised of specific classes of molecules which organize themselves into structures and chemical pathways. Hence, in order for life to emerge, order must arise from chaos. The next step of complexity occurred as certain components of the primordial soup came together, forming the primitive cell or protocell. One of its hypothesized features is a crude form of metabolism that allows the protocell to exchange energy and material with the environment. At the center of this protometabolism, many believe, is a property called autocatalysis.
Autocatalysis, in its simplest form, can be represented by this equation. A reacts with B, forming two copies of itself. This positive feedback, often avoided in traditional chemistry, opens up all sorts of behaviors like oscillations and wave formation that regulates modern-day biology. In fact, cell and DNA replication are also forms of autocatalysis. My research focuses on one of the oldest prebiotic chemistry reactions, called the Formose reaction. Originally, scientists focused on the Formose reaction for its ability to make ribose, which is the sugar component you find in RNA. However, I find the possibilities for oscillations and other complex behaviors more interesting. Researchers are still discovering new aspects of this classic reaction first discovered in the late 1800s. And I think that's what makes origin of life such a compelling puzzle. There's always a new angle.
Metabolites, like the products of the Formose reaction, would be lost to the environment without some form of encapsulation. Modern cells rely on the cell membrane as a physical barrier for this separation. It is proposed that protocells also possessed a membrane, albeit a simpler one.
Cell membranes in modern life forms are made of phospholipids and membrane proteins. These molecules which require enzymes to form, would have been too complex to exist when primitive life first emerged. It has been proposed that the first membranes were composed of fatty acids, which can wrap in spherical bilayer structures known as vesicles. Fatty acids are chemically simpler than phospholipids. They can be produced abiotically and can self-assemble into vesicles under the right conditions. In my research, I hope to find a mechanism through which primitive cells could have had continual access to the nutrients required for membrane growth to occur. This will help to create a minimal synthetic cell that can grow and divide, helping us to understand how protocells could function without complex cellular machinery.
Besides vesicles, there is another method of encapsulation that has caught the interest of researchers.
Coactivation is the formation of membraneless liquid organelles, where a condensed phase forms distinctly separate from the dilute phase that the solutions are prepared in. Predominantly, we find this is driven by electrostatic interactions whereby oppositely charged ions come together to form these condensed phases, meaning we can replicate these using peptides and RNA. Interestingly, when we condense these down, we actually find that they are capable of driving biochemical interactions, making them favourable for studying protocells. So my research focuses on studying amino acylation using these biochemical reactions that we find happening in coacevates. Amino acylation is the body's way of producing proteins, meaning it is essential for life to occur. So in the body, we find that amino acylation is performed by the ribosome. What the ribosome is just a complex mixture of peptides and RNA. Therefore, we can mimic this using coacevates, which we know to have been able to form in a prebiotic scenario.
Understanding the components of the protocell, however, provides only a portion of the puzzle. Where these chemical processes occurred is just as interesting as the processes themselves. In order to support a variety of reactions, a landscape should possess a diverse range of pH, temperatures, and minerals. A hydrothermal field is an example of such an environment.
Hot springs form when rainwater percolates down through porous cracks and fissures in the underlying rock. This water is then heated by an underlying magnetic source, which causes convection and upwelling of fluids. Eventually, these fluids erupt at the surface as mineral-rich hot spring fluids. As it cools down, it evaporates and precipitates out minerals. Some of these minerals rapidly entomb and capture this life, causing it to fossilize rapidly, so you end up with fossils of microbes. Some fossils found in the ancient rock record are actually hot spring deposits, and these are in fact the earliest evidence of life on land. For generations, humans have pondered life's big questions, like what is the origin of life? And is there life elsewhere in the universe? These questions are so easy to ask, yet so remarkably hard to answer. Hot springs are dynamic, ever-changing, almost alien-looking environments, and studying them can provide the opportunity to understand how life thrived on early Earth. Studying them could also help with the understanding of the origin of life on Earth, and if there truly is life out there in the universe.
While Michaela studies samples collected from hydrothermal fields, her colleague Luke Stella conducts lab experiments to better understand the relationship between rocks and geochemistry.
Hot springs have amazing characteristics that make them an incredible place to study the potential environment where life may have formed on Earth. One of these characteristics is their complexity. Hot springs have hundreds or even thousands of individual pools that have unique chemistries, pHs, and temperatures. These pools are constantly mixing in with one another, creating unique little environments where the two fluids interact. My research that I conduct at the Australian Centre for Astrobiology is looking at this complexity and how it could help form the chemicals needed for life to evolve on Earth. In these early life hot springs, what minerals precipitate out and form solids on the edge of the hot spring pools can be really impacted by these organics that are delivered from meteorites that are impacting onto the Earth. So the research we're doing here is trying to understand how that early Earth environment might have looked like, And our understanding of that environment is changing every day as more and more research is coming out. But hopefully eventually we'll have a really good model of that first hot spring where life might have formed on the Earth's surface. And with that, we'll get a better understanding of what you need to create life.
For the next 2 billion years, protocells, perhaps having originated from a basic hot spring, would evolve many of the traits shared amongst all forms of life. The third component of protocells, genetic material, stores information in a way that can be passed on to future generations. Early genes from different protocells would mutate, merge, and split countless times, leading up to the last universal common ancestor, or LUCA.
For the first 2 billion years in the history of life on Earth, life was dominated by these single-celled microorganisms called the archaea and the bacteria. And the archaea and the bacteria are what we call prokaryotic organisms, meaning they lack internal membrane structures. That was up until about 2 billion years ago, where we saw the emergence of a new, more complex cell type, the eukaryotic cell. The eukaryotic cell is the cell type of us and all animals, plants, protists and fungi. And really what the eukaryotic cell represents is a new level of cellular complexity in that it does possess internal membrane structures. So the question is, how did this new, more complex cell type emerge? Current models on the origin of eukaryotes suggest that they actually emerged from the merger of the archaea and the bacteria. In these scenarios, an archaea swallowed up a helper bacterial organism, and the bacterial helper organism over millions of years slowly lost its independence from the host archaea and evolved into an organelle that we now call the mitochondria. The mitochondria provided the archaeal host cell with the energy it needed to become more complex. What I study in the lab is a group of archaea called the Asgard archaea. We suspect these are the most likely candidates for the archaeal host cell that swallowed up the ancestor of the mitochondrium. And what we do in the lab is we try to grow these cells in culture so we can start to determine if these archaea have characteristics that are shared with eukaryotic cells to build a time point for the origin of eukaryotes.
From molecules floating aimlessly in space to archaea engulfing bacteria, these leaps in complexity were critical events in defining life as we know it. Access to unique geological landscapes, combined with the wide breadth of expertise, puts the Australian Centre for Astrobiology at the forefront of research into the origin and evolution of life on Earth.
For trying to answer a question this big, you really can't do it from your individual field. You need to work together in a massive interdisciplinary team. Here at the ACA, you get to chat to all different types of scientists from fields outside of your own, trying to figure out that big question: how did life form on Earth? And are we alone in the universe? And it's only at places like the ACA that we can make this possible.